So today we will begin the chapter on vestibular system. As you have already learned on audition, you know the structure and the receptors for audition are situated in the internal ear. But now this is part of again an internal ear where we have some receptors which are functioning for the maintenance of posture as well as equilibrium in our body. Whether the person is in motion or the person is static, we maintain the posture and equilibrium. And this is possible only because of these receptors that are present, and they together constitute the vestibular apparatus. So today we will begin with the and you know, as part of function we have the cochlea, which is a coiled structure, and it is a structure where there are bony canals as well as membranous canals called as the name of labyrinths. So we have bony labyrinths and membranous labyrinth. This is actually a continuation of the same part of the bony and the membranous labyrinth situated in the internal ear. So this sensory organ is very complex in nature, but at the same time very sensitive. We will come to know of these sensations only when there is some abnormality. Otherwise, we are not at all aware of the sensation because it's an unconscious sensation. We never know how we are maintaining the posture and the equilibrium, whether we are static or we are in motion like running, jumping, walking, whatever it is. So, this apparatus or the sense organ help us in maintaining the posture and equilibrium in motion as well as not in motion, that is when we are static. Coming to the structure, we will just learn about the functional anatomy and the mechanism how it gets stimulated. So this vestibular apparatus actually is seated or housed in the petrous part of the temporal bone, where the structure for the audition is also there. As I told, it's all continuous, right from the oval window, then it goes to the cochlea. So this is also part of the same internal ear, and we have structures mainly of two parts. One is a semicircular canals, and there are other two sac-like structures called as a utricle and the saccule. So we take the midline and we have two internal ear on either side. We have three semicircular canals which opens into two sac-like structures which are continuous. So this is a utricle and this is a saccule, and these are the three semicircular canals. As I told you, it's a bony labyrinth with a membranous labyrinth where fluid is filled in both these cavities, the bony labyrinth as well as the membranous labyrinth. The membranous labyrinth is lining the bony labyrinth, so there is a fluid between the membranous labyrinth and the bony labyrinth, which is called as a perilymph. So we have the bony labyrinth which is filled with perilymph. At the same time, the membranous labyrinth is there, which is filled with a fluid called as an endolymph. Both of them are different for some reasons. So the perilymph and the endolymph will be located in such a way that perilymph is situated within the bony labyrinth and the membranous labyrinth, whereas the membranous labyrinth will be filled with endolymph. And endolymph is secreted by the stria vascularis, which I have already learnt along with audition. So the fluid is continuous in all these structures, and it is rich in potassium, whereas perilymph is rich in sodium but not with potassium, and it is produced from the extracellular fluid, and it is similar to the cerebrospinal fluid. So those are the differences between the perilymph and the endolymph. Now these three semicircular canals we'll learn in detail, which opens into a sac-like structure commonly, which is called as a utricle, and in turn the utricle will be opening into another sac-like structure which is called the saccule. So we have the utricle and we have the saccule. This saccule will be continuous with the which structure? Cochlea. So now we say there are two divisions functionally for the structures that are seated within the petrous part of the temporal bone. The semicircular canals along with the utricle and the saccule will be concerned with the maintenance of the posture and equilibrium, whereas the cochlear part is concerned with audition. So we have two divisions of the 8th cranial nerve, which is called as a vestibulo-cochlear nerve. The vestibular division of the 8th nerve will be innervating these structures, and the cochlear division will be innervating the cochlea. So there are two functions for this particular cranial nerve. And now we will go into the details of this semicircular canals, where these canals are actually lying in such a way where they are perpendicular to each other. Follow. And these semicircular canals are named as, one is the anterior one, otherwise called as a superior one. Then we have another which is a posterior one, or otherwise it can be also called as the inferior one. And we have another canal which is called as a lateral one, or it can be otherwise called as a horizontal one. So we have the anterior or the superior, posterior or the inferior, and the lateral or the horizontal. So accordingly we have the same structures on the right side also. But there is something different where when they function together, because we have on either side. The vestibular apparatus is present on either side. And these canals will be presenting its position in such a way that if a person inclines the head 30 degrees in the forward direction, they all will be directed in such a way that the anterior canal will be directed outward and anteriorly. Follow. It will be directed anteriorly as well as outward. The posterior canal similarly will be directed outward and posteriorly. And the horizontal or the lateral canal will be lying parallel to the ground. This is how these canals will be directed or oriented when a person inclines the head 30 degrees forward. And we have to understand one more point where this anterior canal on the right side, let us say this is the right side and this is the left side. This anterior canal on the right side will be in the same plane of the posterior canal of the left. So the anterior will be in the same plane as the posterior of the inferior on the left side. Similarly, this posterior, because it is in line in plane with the anterior, similarly we have the same anterior in plane with the posterior of the right side. Is it clear? So they are in the same plane. This is specially mentioned because they always function together, not on the one side. So here again when we take the lateral one or the horizontal one, they will be in the same plane. So this is how it looks like. The anterior of the right in plane with the posterior of the left, the anterior on the left in plane with the posterior of the right. The horizontal or the lateral will be in the same plane. So whenever they function together, we see that they are just like mirror images. Moreover, the action can be described as being pushing and pulling, or a push-pull fashion. If one side it is a push, we are considering it as a push, the other side will be a pulling effect. We will come to it later. So this is how these canals will be functioning. So this is just an orientation of the canals. Going to more details of the canal, we see that these three semicircular canals are opening into the utricle, the next sac-like structure. So they have two free ends, which we will consider as 6 openings through which these 3 canals will be opening into the utricle. But there are only 5 openings. This is because the anterior and the posterior together will be sharing a single opening. So these 3 canals will be opening into the utricle by means of 5 openings and not 6 openings. So there are 2 free ends for all the semicircular canals, but one free end of all will be having a dilated structure which is now called as the ampulla. So each canal, the free end is there, one end will be dilated. Again for the horizontal canal we have the free ends, one end which will be dilated. Again for the posterior one we have 2 free ends, one will be dilated. It is this dilated portion which is now called as a ampulla. So now we see there are 3 ampullae on either side. It is the sensory receptor which is now seen in these dilated ampullae and is known by the name of crista ampullaris. So the sense organ is now called as a crista ampullaris. Now when we go into the details of this structure, there is a sensory receptor. For an easy learning, we will consider the horizontal canal so that it will be easy to draw and learn. So let us consider this as the midline where we have the horizontal canal with the ampulla at one end, and here again we have the semicircular canal with the ampulla at one end. It is a thickening of this epithelium of the membranous labyrinth which give rise to the structure of the sensory organ. That is what is called as a crista. Together named as crista ampullaris because it is located within the ampulla. Now this structure is actually seen like a dome shaped structure which is a thickened epithelium of the membranous labyrinth. So there it is actually the tall columnar epithelial cells which are also ciliated. So in short they are epithelial cells, but in between we can see there are the supporting cells also. So actually these epithelial cells which give rise to the sensory receptor are actually modified neuronal cells acting as a receptor. So they are epithelial ciliated cells, actually modified neuronal cells acting as a receptor cells. And there are in between the supporting cells or the sustendicular cells also. So these are situated within the ampulla, and you should understand there is something on top of it where there is a gelatinous structure called as a cupula. So there is a gelatinous structure overlying the cilia of the tall columnar epithelial cells which is called as a cupula. And this structure, although I have drawn like this, it will usually be filling the whole of the ampulla. So it completely obliterates itself, filling the whole of the ampulla. So this is a crista ampullaris. And the detailed structure is in such a way that we have the epithelial cells which are ciliated. On top of it there is a gelatinous material called as a cupula. And it will be totally filling the whole of the ampulla, so that whenever there is a fluid motion of the endolymph, usually it will obliterate its flow at the level of the crista. Is it clear so far? Now, again going to the details of these cells, we should know something about this cilia. Because when we consider a single epithelial cell, which is a neuronal cell, we know there are cilia present in these epithelial cells. They are actually an extension of the cytoplasm giving rise to hair-like structures. So these hair-like structures will be in such a way that there is one tall cilium with shorter cilia directed towards one direction. This is how it looks like. So we see that there is one tallest cilia which is now called as a kinocilium, and the shorter ones which are decreasing in the height are called as a stereocilia or stercocilia. Both are the same because some textbooks say it is stereo, some say it is stercocilia. So these are the cilia which are seen, or the hair cells which are seen nearer the kinocilium, decreasing in height. But at the same time they are connected by membranous structures which are called as tip links. Now when you learn about the audition, there also it is the same hair cells which are acting as the receptors. So these are actually mechanoreceptors where a distortion or a displacement of the hair cells will be stimulating or will be giving rise to an action potential. So when we learn about the mechanism, we will come to know what is the stimulus for these receptors. So coming back again to the kinocilium and the stereocilia, we know that there is one tallest kinocilium. And whenever the movement of the cilia occurs, if it is towards the kinocilium, it always results in a depolarizing signal. And if these cilia are moving away from the kinocilium, it always results in a hyperpolarizing signal. So when we consider this crista ampullaris, where we have the receptor within the ampulla, there are 3 ampulla because there are 3 semicircular canals, and there are 6 total because there are left and right semicircular canals. And here, as I told you, the pairs which are working in the same plane are called as the synergistic pairs, and it will be in a pull or push fashion, and it is also a mirror image. Why I say it is a mirror image? Because when we consider the right semicircular canals with the crista or the receptor, we see that it is towards the utricle that the kinocilium will always be directed. Followed. Always the kinocilium will be directed toward the utricle, but for a better learning and understanding, you just imagine that the kinocilium will be always towards the midline. That will make learning very easy. This is because always the kinocilium, because the semicircular canals open into the utricle, the kinocilium is also towards the utricle. So when we consider 2 semicircular canals on the left and the right, the kinocilium on the right side will be just towards near the midline. Here also it will be nearer the midline. So now when we consider the neuronal signal, what will happen? If this is moving, both are moving to the left side, what will happen? On the right side it is moving towards the kinocilium, but on the left side it is moving away from the kinocilium. So the signal on the right side will be the depolarizing one, and the signal on the left side will be of a hyperpolarizing one. So the planes in which 2 canals will be working will be in such a way that the movement of the head of the body, whatever it is towards one side. So that side may be producing an excitation that will come to later, will be producing an excitatory signal. At the same time, it is an inhibitory signal on the opposite side. That is very important. This is the basis for learning how the mechanism of the semicircular canals or even the receptors of the utricle and saccule works. So basically, this is the principle behind the signal transmission from these receptors. So now we will go to the mechanism of action where this happens. So finally, when we learn that it is the hair cells which are acting as the receptors, they are mechanoreceptors, and the distortion of these hair cells are responsible for producing a signal. Now we see that these stereocilia and the kinocilium, because of their attachment to the tip links, if it is towards the kinocilium that the movement occurs, it produces a depolarizing signal. This is because at the tip of these cilia are potassium channels. So whenever it moves towards the kinocilium, it is in such a way that these potassium channels become opened, and potassium will enter into it. Even calcium also enters the cilia, resulting in a depolarizing signal. And the innervation of all these epithelial cells will be by the fibers coming from the vestibular division of the vestibulocochlear nerve. Finally, they will be going to the vestibular ganglion, or otherwise called as a Scarpa's ganglion. That forms the first order neuron, because all sensations are through a 3-order neuronal pathway. So the first order will be the Scarpa's ganglion or the vestibular ganglion. That way it goes. We will learn about the pathway later on. So this is how it occurs, and when there is a depolarizing signal that occurs, which is due to the change in the hair cells, when it reverts back, it finally gives rise to the closure of the potassium channels, and the depolarization signal will be stopped. Now continuing with the structure of the receptors present within the utricle and the saccule. Those are also receptors which are functionally the same. That is, the mechanism is the same, where the hair cells will be stimulated. So the structures in the utricle and the saccule will be in such a way that the direction of the hair cells are different in the utricle and the saccule. Moreover, there are also slight differences. Here again it is the epithelium that is thickened to form with in between we have the supporting cells, and these are also ciliated just like the other one. And we also have the gelatinous material, which is a cupula. But another important difference is that this cupula or the gelatinous material is studded with very many calcium carbonate crystals. So this gelatinous material on top of this hair cells of the ciliated epithelium is studded with calcium carbonate crystals. They are also called as otoliths, and hence this is otherwise called as a otolith organs. These are also called as otolith or otoconia. So this is one important difference where this cupula or the gelatinous material is studded with calcium carbonate crystals, and so now we see that the endolymph is of high specific gravity compared to the perilymph. And because this cupula is now studded with this calcium carbonate crystals, that will add on to the specific gravity, which is almost twice that of the endolymph, and hence this is always under the influence of the gravity. So these receptors, that is the receptors present in the utricle and the saccule, are always under the influence of gravity because of this high specific gravity contributed by the calcium carbonate crystals on top of the gelatinous material or the cupula. Followed. And another important difference is that the arrangement of these receptors in the utricle and the saccule are also different. In the utricle, it is arranged in a horizontal manner, so the direction of the hair cells will be vertical. Whereas in the saccule, it is arranged in a vertical manner, so that the hair cells will be in a horizontal direction. So that is the difference here. So the direction of the hair cells will be different in the utricle and the saccule, where the hair cells are directed vertically because it is placed horizontally, and here because it is arranged vertically, hair cells will be in the vertical direction. Now we will come to the stimulus where these receptors will be functioning. When we learnt about the semicircular canals, we know that in the midline we have all the three ampulla with the crista, isn't it? Filling the whole of the ampulla. So they are erect in the standing position of any individual. Whereas in the same way, when we consider these two, they are in these positions or oriented in these positions in the utricle and the saccule in a person who is static. But this is always under the influence of gravity. For the same reason, there will be a continuous stream of impulse that is being sent from these receptors to the brain because of the influence of the gravity, appraising the brain about the position of the body with respect to the gravity. Whereas these structures in the ampulla, they get stimulated only when there is a change in the position of the head in relation to the body. So we see that this is a tonic receptor where the otoconia are acting as tonic receptors with a continuous stream of impulses even during rest, whereas this becomes stimulated only when there is a motion that happens within the body or of the head in relation to the body. So this is acting as a phasic receptor. So these are the tonic receptors and these are the phasic receptors. But acceleration is the main force of stimulus for all these receptors. So the acceleratory force for this crista in the ampulla always will be angular or rotational. So the acceleratory forces can thus be divided into angular and rotational for the crista ampullaris, and it is the gravitational for the otoconia. And this is a tonic receptor and this is a phasic receptor. But both are stimulated by the acceleratory forces. Now when we consider the angular acceleration, when a person notes this or head by saying yes, it goes in the anterior-posterior direction. So that produces an angular acceleration that will stimulate the semicircular canals or the receptors in the semicircular canals. Even if the person says no by nodding the head left to right, that is also another angular acceleration that will stimulate these receptors in the semicircular canals. Even otherwise, if the person just turns his head to the left or right, touching the shoulders, that is another form of angular acceleration that will be stimulating the receptors of the semicircular canal. Rotational, as the person spins the head, that also will give rise to the same type of angular acceleration which will stimulate the receptors of the semicircular canal. But here, the otoconia, which is always under the influence of the gravity, we are always aware when we are inside a lift. We are just aware because even if the lift is going up or down, we will be sensing it and we say the lift has started up, or we say the lift is going down, or the person as is jumping up from a platform or going up. Those all will be, the person will be knowing that when the plane is being taken off, we know it. When the plane is going to land, that also is being sensed. So all these are only because of this otoconia. So in spite of the person being stable, they are aware of the motion that happens due to the motion that happens only because the person is as a whole is being moved in a particular vehicle or whatever it is. Even otherwise, we sense it. So we become aware of these senses when there is a motion that happens. Similarly, here also we become aware. Otherwise, when we are just walking, sitting, lying or running, we maintain the posture and equilibrium. That's why I told you it's an unconscious sensation. Only when it becomes abnormal, we become aware of it. Just like the palpitation, we become aware of the heart rate. When we become dyspneic, we become aware of the respiratory system. Like that, this is unconscious sensation which become conscious when there is some abnormality and it disturbs the posture and equilibrium. Now we will come to the mechanism by which these are stimulated. So always remember, these receptors are stimulated by acceleratory forces. We will just learn how the semicircular canals are in action when there is an acceleratory force acting upon it. So here you have to understand one point where the endolymph has an inertia always. So whenever there is a rotation or movement of the head or the body, there is a movement of the endolymph and the perilymph, because the bony labyrinth also will be moving along with the motion of the head. So when there is a motion that happens in the bony labyrinth, it usually will produce a movement of the endolymph, because that forms a basis for the tilting of the hair cells. That's a basic mechanism even in audition, because there is a basilar membrane which fluctuates because of the motion of the endolymph. On top of it we have the, what's the structure in the audition? Organ of Corti. So there again it is the hair cells which are stimulated by the movements of the membrane. And again when we go to the mechanism of hearing, there are so many aspects of hearing like the pitch discrimination, etc. Localization of sound, so many things. But basically the hair cells become displaced indirectly because of the situation on the basilar membrane, and it is the endolymph that produces the displacement. Here again, when there is an acceleratory force that comes into action, it will produce a mechanical tilting of these hair cells, and because they are neuronal cells acting as receptors, they are polarized cells, they undergo depolarization, and there is a rule whereby the movement of the hair cells, if it is towards the kinocilium, always results in a depolarizing signal, and if it is away from the kinocilium, it results in a hyperpolarizing signal. So now we learn how this comes into play. Let us consider the horizontal canals first. So we have the crista here on either side in the ampulla, isn't it? So in a person who is upright, it is erect. Let us say that this particular person is turning the head on to the right side, isn't it? When the person tilts the head to the right side, what will happen? Bony labyrinth also moves to the right side, isn't it? Now we have the cupula which was upright at rest. What will happen when the bony labyrinth moves to this side? Cupula will also be displaced to the right side. So here the cupula will be going to this side. Here again this goes to this side. That is what is going to happen. But because of the inertia, the endolymph will be lagging behind, and the movement of this cupula will be in a direction. The movement of the head will be to the right side, whereas because of the inertia of the endolymph, the displacement of the crista will be in the opposite direction to begin with. So here the head is rotated to the right side, but because of the inertia it will be standing behind, and so what happens here? This is actually, instead of going to the right side, it will be displaced in this direction, isn't it? Is it clear? So because of the inertia of the endolymph, the erect crista now will be at the initial part. Because of the inertia, they are displaced in the opposite direction of movement of the head. So when we consider the movement of the head on the right to the right side, this cupula moving to the left side results in what type of signal here? As I told you, always consider towards the utricle, you can consider it as the midline. So kinocilium is here and kinocilium is here for the left one. So here when the kinocilium is moving in the, or when the motion of the endolymph is in the opposite direction of motion of the head, here what happens to the stereocilia? Is it going towards the kinocilium, or it's towards the kinocilium on the right, resulting in a depolarizing signal on the right, whereas on the left it is going away from the kinocilium, and the left it results in a hyperpolarizing signal, isn't it? So the head is being rotated to the right side, where it results in a depolarizing signal going from the right side of, or right semicircular canal, but on the left side it is an inhibitory signal. But after few seconds, the momentum will be catched by the endolymph, and as a rotation continues, the inertia will be lost and the endolymph will be in the direction of motion. So the displaced crista will now come back to its original position. And when the movement continues or the rotation is continued, there is nothing happening. So there will be signals equally transmitted, or there is no stimulation that happens to the brain from either of these crista. Followed. And just when the rotation is stopped, what will happen? Again the same thing happens because of the inertia of the endolymph, and the displacement of the crista will now be in an exactly opposite way that was there in the beginning. So now here the displacement will be in this opposite direction because the displacement to the right has been stopped. So it will be again in the opposite direction, and here instead of the kinocilium, the stereocilia directed towards the kinocilium, it is going away from the kinocilium on the right side, and here it will be towards the kinocilium on the left side. So it becomes an inhibition on the right and excitation on the left at the end of the rotation. Followed. So to begin with, right side is the motion of the head. So to begin with, again it is excitatory signal that goes from the right side. The placement is, that's why I told you it's always a push-pull fashion, always a mirror image. Followed. And this is acting in the same plane, semicircular canals. If the angular rotation is to be functioned by the anterior right, the posterior left will be in the same plane. They together will be functioning. Similarly, the right posterior and the left anterior. That is why they are called as a synergistic phase. So this is what is happening, because we always will not be moving in the, if it is the same direction or vertical, means there is nothing happening. If there is a motion or movement of the head to one side, that has to be informed to the brain that the person is moving to the right side. So the signal that goes to the brain will be excitatory, or it is a depolarizing signal reaching the brain. Same time it is a left that is going, that's inhibitory. At the end of the rotation, it becomes a reverse. So both the signals are simultaneously sent to the brain, where the brain will be apprised of these signals, and the person will know that I am going either to the right or to the left. So this is the importance of this mirror image or the pull-push fashion of this functioning of these crista in the semicircular canals or the ampulla. So when we consider the other one, there is a utricle and the saccule. As I told you, when a person moves up the lift, which will be functioning? When we are going up the lift means, what should be the direction of the hair cells? Which should be horizontal. Where is it placed horizontal? Where are the hair cells in the horizontal position? It is placed vertically in the saccule. So saccule is a one, isn't it? So if you are attentive, you will answer which is a one. If I have said it wrong, you will correct it. Utricle, it is arranged horizontally. Hair cells are vertical. So it is a horizontal movement that will be stimulating the horizontal cells. Sorry, is the utricle. And now if it is a lift means the hair cell should be in the horizontal direction. So it is the saccule that will be stimulated. So never go wrong. In the utricle, it is arranged in a horizontal position, so that if it is placed horizontally, hair cells are vertical. Because if it is placed like this, hair cells are vertical. So it will be moving in this direction. When a person is moving in a car or a train. So that is why when we go in a ship, and especially if the sea is very rough, what will happen? All sorts of movements will be there. And if the person is having a very sensitive vestibular apparatus, that creates vomiting sensation and dizziness. We will come to it later on. So these movements, the vertical and the horizontal arrangement of these receptors separately in the utricle and the saccule is also with a purpose, where along with the gravitational pull by the autoconia, the person will be made aware of the motion that happens or the acceleration of the body that happens, whether it is in the horizontal direction or in a vertical direction. Similarly, these cells will be stimulated to the maximum until we attain a posture or equilibrium. When a person walks uphill, how will he walk or she walk? Uphill. What is the posture adopted when a person is going uphill? Person will be stooping forward, that is to catch equilibrium. And here the stooping will be correspondingly equal to until the person maintains equilibrium, so far where these hair cells will also be tilted. And the tilt will be in such a way that until the person maintains equilibrium, that tilt will also be proportionately more. If a person is going downhill, he will be walking like this. This is also with a purpose, where these hair cells will be helping us to maintain the equilibrium, because they are always under the influence of gravity. That's why they are called as tonic receptors. The other one is called as a phasic receptor. So now at least you have understood the mechanism, how this functions. Now we will see some of the PPT here. And this is the structure of the external ear, the external auditory meatus. Now it directly enters the cochlea. That's a portion which will be helping in the audition. Now you can also see in the same part of the temporal bone where we have the three semicircular canals, the anterior, superior, posterior, inferior, and the horizontal one. Okay. Now coming to the next slide, here the membranous labyrinth is being shown. You can see in the enlarged form where we have the three semicircular canals. This is a lateral semicircular, this is a posterior, this is a posterior, and this one is the anterior. And they end up in the ampulla, which is a dilated portion. These are the three dilated portions. And here is a oval window which is related to audition, but you can see the vestibule here. At the same time, you cannot see the utricle and the saccule. Here we can see the vestibular receptors which have been shown in the form of a cut portion or a section. So we have the 3 ampullae where we have the receptors, which are the crista, and we have also receptors of the utricle and the saccule seen here in the utricle and the saccule, and the 2 divisions of the cranial nerve, the vestibular ganglion. That part which goes to the audition is the cochlear division. This is the vestibular division. This again showing both the bony labyrinth and the membranous labyrinth with all the canals. Again a cut section showing the bony labyrinth and the membranous labyrinth. So the whole structure of the internal ear is seen in this diagram. Here comes the structure of the, which is the structure, it is the otoconia or the otilis which was seen here. So it is actually the receptor seen in the utricle and the saccule. Since it is placed horizontally or vertically, the hair cells are seen in the vertical direction. So they are placed in the horizontal way, which is actually the receptor which is seen in the saccule. And if you go back, we can see these are the epithelial cells which are acting as the modified neuronal cells, which are the receptors with supporting cells in between. And you can see the cup-like structure seen on the base, they are the nerve fibers innervating these receptors. And you can see the membrane also here, the gelatinous membrane, on top of which we have the otoliths or the calcium carbonate crystals. Here again you can just see the location of the receptors in the 3 semicircular canals. These are the 3 cristae, and these 2 are the receptors in the utricle and the saccule. And this is in relationship to the position of the head. You can see this is a horizontal semicircular canal here, which is shown, and this is in the right anterior, this is the right posterior. So the right anterior and the left posterior will be in the same plane, the left anterior and the right posterior in the same plane, and we have the horizontal canals or the lateral canals in the same plane. This is just to show the position of the cupula in the upright position and the displacement when there is an acceleration. So here it is in the upright position, B it is a displacement of the cupula, and when we consider this is an angular acceleration, the flow of the lymph also showed here. So this is actually towards the utricle, where the signal transmission will be in the form of a depolarizing signal. So correspondingly the rotation and the position of the canals have been shown here. So here right posterior is a left posterior, right anterior, left anterior. So the planes will always be there, they are acting as a mirror image or push-pull action. Now it's an enlarged diagram of the crista. You can see it is fully filling the ampulla, obliterating the flow of the endolymph. You can see the cilia also on top of this is slightly a cut section which is shown. We have the epithelial cells here, the nerve fibers, and also the gelatinous portion. This is another diagram where you can see the crista filling the whole of the ampulla, but you can also see the kinocilium and the stereocilium, where the kinocilium is the tallest one and the height of the cilia goes on increasing as we reach the kinocilium. Here you can see the stereocilia when they bend towards the kinocilium, there is a depolarizing signal. It is due to the release of an excitatory neurotransmitter which can be either aspartate or glutamate, and so the spikes will be 160 per second. It's an excitation. When it is at rest, it is only about 90 spikes. It is upright. Here again it is going, the channels get closed when the cilia move away from the kinocilium, and see the number of spikes are 20 spikes per second. It's actually the release of the neurotransmitter will be inhibited. Here this will show the tipplings. And you can see in the upright position the spiral-like structure which is a tippling, and you can also see the potassium channel. As it moves towards the kinocilium, there is a stretch of this tippling, opening of the potassium channel, wherein calcium also enters along with the potassium, giving rise to the depolarizing signal. And finally, when it gets opened, after that, the stretch on the tippling will be taken away so that it again becomes relaxed and it gets closed. we said that a myosin filament is related to this mechanism. Now we will see how it works. So this is the hairstyles which we have with the tallest kinocilium and the stereocilium. Nothing more is seen here, and you can see the attachment of the cilia. Here, when it is deflected to the left towards the kinocilium, there is the opening of the channels and it results in depolarization. Again it comes back, especially when the inertia of the endolymph is over and the movement of the endolymph will be in the direction of the motion, coming to the upright position. Again, when it moves in the opposite direction, it is away from the kinocilium, resulting in an inhibition. This is again another diagram of the crista, the nerve fibers, etc. Again the movement of the kinocilium and see the direction of the endolymph resulting in the movement of the cilia towards the kinocilium results in a depolarizing signal. Whereas when the endolymph is moving in the opposite direction, it results the movement of the cilia away from the kinocilium, resulting in a hyperpolarizing signal. This again is to show the position in the resting state is 90 on either side. But when there is an excitation to one side, it results in about 180, whereas in the opposite side it goes to an inhibition with 10 spikes per second. So I told you the position of the receptors in the utricle and the saccule are different, and they are tonic receptors. The utricle, where the receptor is placed in the horizontal, because it is placed in the vertical, it gets stimulated by horizontal acceleration. In the saccule, it is in the vertical acceleration because it is kept in the horizontal way. This is again another diagram showing the receptor in the utricle and the saccule. This will again show the movement of the cilia. See, head motion to one direction, whereas the movement of the cilia will be in the opposite direction. Head motion again on either opposite direction, resulting in a hyperpolarizing signal. Always the movement of the endolymph will be in the initial part opposite to that of the direction of motion of the head. So this is just to show that whenever we are having that maintenance of the posture, when a person climbs uphill, the displacement of the cilia will be to that limit until that person maintains the equilibrium. So the displacement of the cilia goes on until the person can maintain the equilibrium. So depending upon the steepness, when the person goes uphill, the person slopes forward. If the person is going downhill, he will always be leaning back. So this extent of the leaning forward or backward, as the case may be, will be depending upon the extent of the cilia also, which gets displaced until the posture and the equilibrium is maintained. This is just to show the movement of the endolymph in relationship to the movement of the head. See here, this is prior to rotation. We have the semicircular canals where the crista are upright, and so the stimulation is the same from either side, or there is no inhibition because the spike or the potentials that are being sent to the brain is equal on both sides. Now you forget about the eye movements for the time being. We'll just focus on this part. So now there is a head rotation to the left side, where we see the movement of the endolymph due to the inertia, and the initial phase will be in the opposite direction to the rotation of the head. So what happens here, because of the inertia in the opposite direction, here it is towards the midline or towards the kinocilium. So it is in the direction of the rotation of the head, that means towards the left side. Here in the opposite side, what we see is it is away from the kinocilium. So there is an inhibition, whereas here there is an excitation. So at the beginning of the rotation, to which direction is the head being rotated, there will be an excitation, at the same time an inhibition in the opposite semicircular canal and the receptor, which is in the same plane. So once the rotation is continuing, the inertia will now catch momentum and the crista will come to the normal position, so it becomes upright. Again it is the same like the resting position. After that, at the top edge of the rotation, what happens is exactly opposite to what had happened in the beginning of the rotation will happen here, where initially this was stimulated, now this is inhibited, whereas this which was inhibited will now be excited. Now this movements of the eyes will be learnt later. So now we have finished the functional anatomy of the vestibular apparatus and the detailed structure of both the type of receptors in the vestibular apparatus and moreover the mechanism of action. We will now go to the pathways as well as the function of the vestibular apparatus in the next class, and further we will learn about the abnormalities like the applied aspects and also how the vestibular function is being tested clinically. So today we will stop here.